presenters a thumbs up when there is a 1 minute warning to the end of their talk. And then we do have 2 minutes for questions in this format at the end of the talks as well for everybody. So please think of your questions. So I will get us going here and introduce our first speaker, which is Jasper Greysson-Wong. Jasper, if you would like to share your screen and remove your mute, and then you can begin when you're ready.
Okay, hopefully you can see that. All right. So hi, my name is Jasper, and I'm going to be talking to you guys today about my PhD work using zebrafish to model a blood vessel disease called RASA1 capillary malformation and arteriovenous malformation. So first, we need to understand what RASA1 is and what CM-AVM is. So to dive into the two aspects of capillary malformation and arteriovenous malformation, we know capillary malformations are dilated beds of capillaries in the skin, and they look like, hopefully you can see my mouse, this on the left side of the slide, so also known as port wine stains. I think it was 95% of RASA1 CM-AVM patients have capillary malformations, and the rare presentation of CM-AVM is arteriovenous malformation. And an arteriovenous malformation is a direct connection between artery and vein, which would normally be interceded by a capillary bed, but this fast flow lesion is particularly dangerous because it can rupture because it's not properly supported by mural cells. And so depending on where it's developed and if it ruptures, it can be quite dangerous. So AVMs are present in about 1/3 of CM-AVM patients. So we have these two presentations, and we know that this disease is caused by RASA1, but we don't know what the role of RASA1 is in blood vessel development. The only thing we know about RASA1 is that it's a RAS inhibitor, so it functions as it functions by binding RAS proteins and forcing them to hydrolyze their own phosphate group and turning them into their inactive RAS GDP-bound form. So we need to investigate the role of RASA1 in CM-AVM, and we've decided to do this using zebrafish as our model. So why are we using zebrafish? There's a couple of reasons why zebrafish are so useful in modeling blood vessel diseases. And one of these reasons is because the embryos are transparent. So this makes them easy to image, especially live embryos, and they develop really rapidly. So by 1 day post-fertilization, they have a full circulatory system. And 2 of the transgenic zebrafish lines I use in the lab are a green fluorescent blood vessel transgenic line. So a transgenic line is a tissue-specific promoter that's driving a fluorescent protein. So this allows us to visualize the live development of blood vessels in our zebrafish embryos. The other transgenic line, which is my favorite, is GATA1 DS red, and it is red fluorescent red blood cells. It's really interesting to visualize the blood circulating through your embryos. So we are going to model RASA1 mutations in our zebrafish. And what do we find? We see that RASA1 mutants by 2 days post-fertilization or 48 hours post-fertilization have really large vascular malformations at the posterior of the tail versus their wild-type counterparts where this is not seen. In using confocal microscopy, we can further look at these malformations, and we see that they develop at the back of the tail and predominantly seem to affect the vein. So I wanted to further understand the role of AVMs in this vascular disease. And there's an interesting hypothesis floating around in the literature talking about how the AVM initiation is genetically driven, but the progression of an AVM, so the worsening of the AVM, is actually driven by flow defects from this vascular malformation. So we wanted to see if flow was affected by the formation of this cavernous vessel at the back of the zebrafish tail. So we quantified blood flow velocity throughout the tail. So in the dorsal aorta and wild types, you can see nice laminar flow, and then it becomes a little bit more disrupted in the caudal vein because of its branched structure. In mutants, it follows about the same pattern early on, but by that 2 day time point when we see that really large cavernous vessel, we see blood flow is really slowed down right at that malformation. So I wanted to understand if this had any transcriptional effects. So I stained for CLIF2A, which is a flow responsive transcription factor, and I saw that in RASA1 mutants, there was a decrease in CLIF2A staining in the trunk. So to summarize what we've seen so far, we know that RAS1 mutant zebrafish embryos develop arteriovenous malformations at the back of their tails, and that these AVMs disrupt flow as well as flow-sensitive signaling, as we saw with the CLIF2A staining. So next, I wanted to dive into what drives AVM formation on a cellular level. So one of the main hypotheses about why an AVM would develop is if artery and vein specification is altered, allowing for arteries and veins to fuse. So we looked at two key specification, two main specifiers of artery and vein. So for artery, I stained for FRNB2A, and for vein, I stained for FB4. So we didn't see any obvious differences between our mutants and our wild types for either these artery or vein markers, which leads us to think that early artery vein specification is normal. So one of the other hypotheses I had was that maybe there's an overproliferation of cells, of endothelial cells in the tail resulting in that cavernous vessel, or that maybe there was weird cell death in the back of the tail, resulting in a collapse of what was normally a honeycomb structure into that large vascular malformation. So I stained for a cell proliferation marker and a cell death marker, and I didn't see any changes in cell death and cell proliferation. So this made me think maybe it's a molecular pathway that's being altered, and there's a molecular pathway that we know is altered in different forms of AVM, and it's the MEK-ERK pathway. So I stained for phospho-ERK, which is the readout of active MEK-ERK signaling, and we saw an upregulation of MEK-ERK signaling in the vein, which is really new. No one's ever seen specifically venous upregulation of MEK-ERK signaling in RASA1 CM-AVM. So I wanted to make sure that this was important in the development of those vascular malformations. So I used a MEK-ERK inhibitor called SL327. And when I apply SL327 to RASA1 mutants, we see a rescuing of that vascular malformation. So this tells us that MEK-ERK signaling, the upregulation of MEK-ERK signaling in the vein is really important in RASA1 AVM development. So what is the importance of MEK and ERK signaling, and why would upregulation of MEK-ERK result in the formation of arteriovenous malformations? So there's two main hypotheses that I'm still working on. And one is that even though we know artery and vein specification early on isn't affected, we think perhaps that downstream MEK-ERK signaling in the artery might impair the maintenance of artery and vein identities. And so even though the early specification is normal, perhaps later the artery and vein become confused and are able to fuse and form that large malformation. The second hypothesis is that this vessel bed where we see the malformation forming is an active bed of sprouting vessels at the time point where the AVM forms. And we know MEK-ERK signaling is important in artery and vein sprouting. And so perhaps if there's an upregulation of MEK-ERK, that the vessels are overly sprouting, that maybe they'll form that malformation. So to summarize what I've been able to find so far in my PhD work, we know that RASA1 mutant zebrafish embryos develop AVMs, that the AVMs disrupt blood flow and flow-sensitive signaling, as we saw with the CLIF2 staining, that early artery and vein specification is normal and there's no change in cell death or cell proliferation, but rather the development of the malformation is driven by venous upregulation of MEK-ERK signaling. So that concludes my presentation, and I wanted to thank my supervisor, Sarah, as well as everyone in the lab. So happy to take questions.
Thank you very much, Jasper. Just while people are formulating your questions here, I'll ask one, and just to let people know, you can ask questions by raising your hand or in the chat box. But I was just wondering why the tail was the area that these seem to form, and was this related to specific signaling or just the hemodynamic effects that occur there in particular in the zebrafish?
Yeah, so we don't know that for sure, like why it's in the tail. It's something that we've talked about a lot. There's a couple of things we've thought about, and one of them being there is a direct connection between artery and vein right at the back of the tail where the malformation forms. So maybe the transition between artery and vein makes that area particularly susceptible to changes in artery and vein specification, but also it's right where the flow completely turns 180. So maybe, yeah, the hemodynamic changes there, or different hemodynamic pressures at the back of the tail make that region particularly susceptible. But yeah, good question.
Thank you. Jennifer has a question. Jennifer?
Oh, it's the question sort of related. So in humans, is there any particular part of the body that these happen more frequently? And are these associated with abnormalities in the development of surrounding tissue due to disruption of oxygen delivery?
Okay, so so the way RAS1 like CM-AVM develops is that the individual with the RAS1 mutation is heterozygous for RAS1, and they need to incur a somatic second hit. So that means wherever the somatic second hit arises is likely the area where that will develop the lesion, because the lesion itself is going to be completely null for RAS1. I think the most common areas for capillary malformations are going to be on the skin, but AVMs is predominantly in the central nervous system and can also form in the brain. And then the second part of the question was about surrounding tissues. So there's work in mouse that shows that it's the knockout of RAS1, or sorry, knock-in of endothelial, so loss of RAS1 in the endothelium, sorry, has the same phenotype as a full embryo knockout. So that tells us that it's specifically the endothelium that RAS1 is required in for normal blood vessel development.
Thank you, Jasper. Really interesting work and amazing pictures you have there of.
Thank you.
All right, we'll shift to our next speaker then, who is Loryn Bohne. Loryn, if you can take yourself off mute and share your screen, and you may begin when you're ready.
Great. Okay, is everything good here?
Yeah, perfect.
Okay. So good morning. My name is Loryn Bohne. I'm a PhD candidate in the Rose Lab. I'm here to talk to you today about the anti-arrhythmic effects of glucagon-like peptide 1 in type 2 diabetic DB mice. So as most of us probably are already aware, atrial fibrillation, or AFib, is the most common arrhythmia encountered clinically. It's characterized by erratic electrical activity in the atria, the upper chambers of the heart here, and resulting in an irregular ventricular response. The mechanisms for how AFib develops are incompletely understood, but we have identified distinct mechanisms that are associated with diabetes in comparison to other comorbidities such as hypertension or high blood pressure. So here's a brief overview of how diabetes can result in atrial fibrillation. It is quite complex and multifaceted with both electrical and structural remodeling aspects. My focus is more on the electrical remodeling side, which is a bit more understudied. In our lab, we use the DB mouse as a model of type 2 diabetes. They have a genetic mutation that causes them to overeat, becoming very overweight, as we can see here in panel A. They also have poor glycemic control compared to wild type counterparts. Last year, we published a paper that highlights their increased susceptibility to being induced into atrial fibrillation compared to wild types. And this was in association with reductions in conduction velocity, which favor reentrant mechanisms and pathways, as well as increases in action potential duration at the cellular level that can favor, sorry, I'm just seeing someone in the waiting room here, that can favor after depolarizations that trigger activity and arrhythmia. So type 2 diabetes is insulin resistant, and the main goal with treating these patients is to increase their insulin sensitivity. One way we can do that is by harnessing the insulin sensitizing power of the endogenous hormone glucagon-like peptide 1, or GLP-1 for short. Many analogs are used clinically. We can see a few of them here on the right. And so my research is to see how these hormones, or specifically GLP-1, can improve AF susceptibility and atrial electrophysiology in type 2 diabetes. Because it has a very short half-life, I wanted to first see, before diving into anything electrophysiological, whether it was actually doing something in our diabetic animals. So I conducted a baseline oral glucose tolerance test where I measure their blood glucose at time 0, and then we administer a glucose challenge through oral gavage and measure their subsequent blood glucose levels every 20 minutes for 2 hours. We can see clearly that the DB animals are quite hyperglycemic compared to wild types. We then assign the DB animals to either saline or GLP-1 osmotic pump groups, and they're also caged with their untreated wild type litter mates. These are then monitored for 4 weeks, and we repeated the blood glucose or oral glucose tolerance test, where we can see that the GLP-1 actually did help their glycemic response to the glucose challenge, just not quite down to the healthy wild type levels. We then moved on to our intracardiac electrophysiology studies. So this involves anesthetizing the animals and inserting a catheter into the right side of the heart, where we can simultaneously stimulate and record electrical activity. So here's a representative of a DB saline animal being induced into atrial fibrillation and then spontaneously reverting back into sinus rhythm. This is then magnified here, and we can see in the surface ECG the fibrillatory baseline lack of P waves and the irregular RR intervals, as well as the erratic electrical activity in the corresponding atrial electrogram. So we do see that the DB saline animals, as expected, showed an increased susceptibility to being induced into atrial fibrillation. The GLP-1 animals showed less susceptibility, but are still more susceptible than wild types. However, we do see that their saline-treated animals are trending towards perhaps a more severe AF phenotype as they have longer AF duration. AERP is the atrial effective refractory period, which is the ability of the heart to be re-excited following a stimulus. And we see that this parameter is normalized with GLP-1 treatment. P-wave duration, which is another ECG marker, which represents the atrial conduction period, is improved with GLP-1 treatment, but is not quite to the same level as their wild type counterparts. This was further investigated using high resolution optical mapping. So this involves isolating the atria from each animal and treating it with a voltage sensitive dye, which we can then excite with a fluorescent light and are able to visualize the conduction across the atrial myocardium. So we can see that initiation of each heartbeat starts here in the SA node, the dark red region at time 0, and then this is then spread across the atria into the left atrium here, which is blue, the latter portion of the conduction period. And indeed, we do see that the DB animals are slower to conduct the activation across the atrial wall and atrial appendages, and there is a modest improvement with GLP-1 treatment. This is consistent with the P-wave finding that we saw earlier. Perhaps most strikingly at the cellular level, we actually see a complete rescue in action potential duration. So these figures were conducted with patch clamp electrophysiology in isolated atrial cells. And so we do see a slight improvement in cell capacitance, which is a marker of cell size. So this indicates that there might be some improvement in atrial hypertrophy with GLP-1 treatment. No changes in the resting membrane potential or upstroke velocity of the action potential, but again, a complete rescue in action potential duration. And this is going to prevent events like after depolarizations and reduce events that will trigger arrhythmia in these animals. So to sum up, we have GLP-1 reduces AF susceptibility in mice in association with improved atrial electrophysiology. And this was seen with an intermediate effect on conduction velocity and a complete normalization of action potential duration. Future experiments will determine the underlying mechanism for these effects, as well as exploring different effects from clinically available GLP-1 analogs. And I'd like to thank my supervisor, Dr. Robert Rose, as well as the members of my lab and the funding institutions that fund our lab as well. And with that, I'd like to open the floor to any questions.
Thank you, Loryn. Very impressive work. This talk is open for questions now, either in the chat box or by raising your hand. I see a first question by Justin.
Hi, Loryn. Great talk, really interesting. So do you care to maybe speculate on potential mechanisms? So do you think it's a cell intrinsic change that's happening with GLP-1, or do you think it's potential changes in or structural changes that may occur in the atria by regulating or by normalizing sort of your glucose control and everything like that? So do you think it's cell specific, or do you think it's the environment around those cells that are dictating these changes?
So I think it's likely a combination. Rarely with metabolic disorders do we see just like one specific thing helping these types of phenotypes. The GLP-1 receptor is known to be expressed in the mouse atria, so it's very possible that the GLP-1 itself is directly influencing the electrophysiology of these cells and the tissue. We also have some preliminary data that suggests that there's also reduced fibrosis in the atria of these animals, which would suggest that potentially there's some action or interaction with the fibroblasts. Also, improving glycemic control is going to improve things like the age-rage axis and improve, like reducing age glycation end products that will also reduce fibrosis. We've previously shown in type 1 diabetic animals that giving them insulin also reduces AF. So we also think that there's probably something to do with increasing insulin signaling and how that can also directly affect the atrial myocytes as well.
Okay, and have you guys looked at whether just treating normal mice with GLP-1, whether that has any effect on sort of action potential or cell-centric sort of components that would, I guess, remove the remodeling aspects, but maybe focus more on the cell signaling?
So we haven't done that experiment yet. I do plan on doing some more acute level experiments with GLP-1 to see if it is affecting the cardiomyocytes directly.
Perfect. Thank you very much.
Yeah, my question was, I think, very similar. I was wondering if you felt this was a specific mechanism to diabetes. I know you used the DB mice, but do you think this would apply the GLP mechanism outside of diabetes?
Very possible. I mean, like, as I mentioned, it could be a direct effect on the heart since the GLP-1 receptor is expressed in cardiac tissue. GLP-1 analogs clinically are also used to help manage weight loss in obese patients. We did not see any changes in body weight in these animals, but they are also a very severe genetic mutation that probably very unlikely that something like GLP-1 would actually help with reducing weight. Doing this in a high-fat diet-induced model would probably answer that question a little bit better because it's just not as severe.
Thank you. Great. Thank you very much, Loryn. We'll keep moving here. Our third speaker this morning is Tristan Dorey. Tristan, if you could unmute and share your slides and go ahead when you're ready.
Great. Can everyone see that? Okay. So today I'm going to talk about sinoatrial node function, more electrophysiology from the Rose Lab. My name is Tristan Dorey. I'm a PhD candidate, and I'll take it away. So the sinus node is the pacemaker of the heart, which initiates each and every heartbeat. It's located in the high right atrium and is responsible for the generation of spontaneous action potentials, the frequency of which maintain normal heart rate. So these action potentials fire spontaneously due to the presence of a diastolic depolarization period, or DD slope. Now, heart rate can be further modified by the autonomic nervous system. Increases in sympathetic outflow to the sinus node due to stimuli such as exercise cause an increase in DD slope and AP firing, resulting in elevated heart rate and subsequently increased cardiac performance. Now, in some conditions, such as hypertensive, heart disease, and heart failure, individuals are not able to achieve a maximal or submaximal heart rate, causing reduced exercise capacity. So here we can see as time and workload increase, those with chronotropic incompetence never reach the same as their healthy counterparts. Now, this is strongly associated with an increased risk of death in this population. Now, despite this, little is known about the mechanisms for chronotropic incompetence in the setting of hypertensive heart disease. So to examine these mechanisms, we used a mouse model of hypertensive heart disease by treating mice for 3 weeks with the vasoconstrictor angiotensin II. This results in elevated blood pressure and a pronounced left ventricular hypertrophy, and then consistent with a chronotropic incompetent phenotype, these mice demonstrate a reduced heart rate response to the sympathetic beta adrenergic receptor agonist isoproterenol. So here we can see 30-second rolling averages where we give a systemic injection of isoproterenol, and the angiotensin II treated animals have a lower heart rate response. Now, to assess if these are due to direct changes in sinoatrial node responsiveness to adrenergic stimulation, we isolated individual sinus node myocytes from both groups and performed a series of patch clamp electrophysiology experiments to measure spontaneous action potential frequency and diastolic depolarization slope in response to isoproterenol. So here we can see a sinus node myocyte and then the attached patch pipette, and we get these recordings of spontaneously beating electrical signals from the myocyte. And then when we superfuse isoproterenol, we get an increase in the frequency. But sinus node myocytes from Ang2 infused mice not only had reduced firing at baseline compared to their saline infused controls, but the change in AP frequency in response to isoproterenol was blunted. This is consistent with a smaller increase in the diastolic depolarization slope, as well as the smaller increase in heart rate in our in vivo animals. To understand how this is working, I'm going to show quickly the beta signaling pathway. So we have the beta adrenergic receptor, which activates adenyl cyclase. Cyclic AMP is generated from that. Cyclic AMP binds directly to HCN channels. These are hyperpolarization cyclic nucleotide gated channels, as well as activation of protein kinase A, which phosphorylates calcium channels. Now, both of these are very important for the genesis and regulation of a diastolic depolarization slope and subsequently action potential firing frequency following sympathetic stimulation by norepinephrine or agonists such as isoproterenol. So we went and looked at these channels individually. So the first one was the HCN or hyperpolarization activated funny current. And here we can see the traces of these measured currents by patch clamp electrophysiology. And in panels B and C, we have the activation kinetics, which are largely dictated by cyclic AMP generation. And we can see in response to isoproterenol, there's less of a shift in the activation kinetics of the funny current in Ang2 infused mice. And because these are dependent for their gating on cyclic AMP, we also measured cyclic AMP generation from the sinoatrial node in both groups. We can see that in response to isoproterenol, we also get a reduced increase in cyclic AMP generation. Now, consistent with reductions in cyclic AMP, as well as HCN channel activation kinetics, we also found that the effect of isoproterenol on L-type calcium current was lower in sinus node myocytes from Ang2 infused mice. This is evident by reductions in peak L-type calcium current, channel conductance, as well as the V1/2 or the smaller shift in the voltage for half maximal channel activation. Now, thus far, these results suggest that the beta adrenergic signaling cascade is blunted in angiotensin II infused mice. So to determine how this was occurring, we used a series of molecular and co-immunoprecipitation experiments. And we identified that a cyclic AMP degrading enzyme called phosphodiesterase 4D or PDE4D was increased specifically in compartments surrounding both beta-1 and beta-2 adrenergic receptors, and that its overall activity in the sinus node of Ang2 infused mice was increased, as you can see in panel D. To confirm if PDE4D was responsible for desensitizing the heart rate response to sympathetic stimulation, we acutely treated myocytes with a PDE4 inhibitor from both saline and Ang2 treated groups. And this PDE4 inhibitor, Rolopram, caused a largely normalized response to the combined application of isoproterenol and Rolopram together. So we can see that AP firing frequency is still reduced before the application of Rolopram, and then after improves AP firing frequency and diastolic depolarization slope. Furthermore, PDE4D inhibition with Rolopram in Ang2 infused myocytes also normalized the isoproterenol stimulated increases in L-type calcium current. So here we have L-type calcium current density over time. The application of isoproterenol, we get an abrupt increase in calcium current and a further increase with the addition of Rolopram in the wild-type animals. But in the Ang2 animals, we see that isoproterenol has a very small increase and then a very large increase following the addition of Rolopram, suggesting that this is critical in regulating this channel activation in response to sympathetic signaling. So collectively, these data suggest that PDE4D plays a critical role in regulating sympathetically mediated increases in heart rate in the setting of hypertensive heart disease, and that this could be a potential target for improving chronotropic incompetence. So here we see that PDE4D likely plays a role in just the healthy state to regulate a pool of cyclic AMP, and thus its activation of HCN channels and calcium channels. But in the setting of hypertensive heart disease, increased recruitment of PDE4D limits this pool of cyclic A, thus blunting the overall response of the sinus node to increases in sympathetic activation. I'd like to thank my lab as well as our funding agencies, and I'll open the floor up to any questions.
Thank you, Tristan. So we're open for questions for Tristan, just while people are digesting all of that and thinking about your questions. Tristan, I just wondered, you had that inhibitor for the phosphodiesterase enzyme there. The sympathetic nervous system has so many actions. Could this be inhibited in humans, translated into a therapy that would be safe, do you think?
Yeah, so PDE4 inhibitors are used clinically. The one that comes off the top of my head is for COPD, is PDE4D is highly expressed in the lungs. So there could be a potential role for repurposing for the use of chronotropic incompetence. PDE4 inhibitors have been used, or PDE inhibitors in general, have been used for increasing cardiac function acutely in the setting of heart failure. But PDE4, as far as I'm aware, has not been used for this application specifically.
Thank you. I see Wayne has a question. Wayne?
Thanks, Justin. Tristan, nice to see you.
Nice to see you too, Wayne.
Very interesting talk. Yeah, I like it very much. So I couldn't help to ask the calcium clock.
Yes.
So you have any comment on that? That's one point. And then another point I want to tell you that, this our friend Andy Marks. So he showed that the phosphodiesterase 4D is actually associated with the renin receptor. So you have any second comment on that?
Yeah, so the two are likely very closely linked. I think that there's probably a clear role for the calcium clock. For those of you who don't know, the calcium clock also plays an important role in regulating diastolic depolarization slope. And so, increases in pKa phosphorylation of the ryanodine receptor potentially, or even compartmentation of signaling to the ryanodine receptor could definitely limit the increases. So, reductions in PDE4 around the ryanodine receptor cause increased leak. And so, if you do the flip side and increase the expression surrounding it, it might cause reductions in the depolarization response. So yeah, it's a very interesting question, and we haven't looked at that. We have a new calcium imaging rig, so potentially pitch this to Rob and see if we can look into that for you.
Thank you.
And we have one question in the chat box from Daniel Vera asking, you mentioned that the PDE4D inhibitor showed positive effects in the heart. Could there be effects in other organs, whether positive or negative?
Definitely, there's always, the potential for off-target effects for, any number of drugs. So I think that, being able to target it specifically to the heart will be critical. We're currently using some adenoviral approaches to see if that we can knock down partially the PDE4 response in the heart. So yeah, to answer your question, it definitely can be. PDEs are expressed to varying levels in a number of different organ systems. So it's something to keep in mind.
Thanks, that's great, Tristan. We have a little bit more time here, so I'll just open it up to everybody for questions to any of the presenters, Jasper, Loryn, or Tristan, if there's any remaining questions. If not, I'd like to thank all the presenters this morning for fantastic presentations, really nicely done. Amazing to see the science that's going on across the Institute. And I will, I see maybe Justin has one last question. Justin, do you want to go ahead?
No, just a congratulatory clapping of the hands.
Oh, sorry, yes, a clap. Yeah.
Just saying good job to all the presenters.
Yeah, great. So I will pass it back to you, Jennifer, then for comments before the break.
Okay, thank you. We're making good time. So thank you to the moderators and the presenters for keeping us on time. And we made it through the whole morning without asking someone to unmute themselves. That's great. So we now have a break. So for those of you who are joining us for the knowledge translation workshop, use this next 5 minutes to go and grab your lunch, and then you're going to come back to this main Zoom room where the workshop will take place. For everyone else, for everyone not participating in the knowledge translation workshop, your lunch break starts now, and we'll finish by 2 p.m. and then start joining back on this main Zoom room at 1:45 p.m. so we can start the oral presentations on time.